Okay, so again, I think we're going to get started. My name is Mike Verneris, and I'm one of the pediatric bone marrow transplant docs here at the University of Colorado. And this is a bit of a special lecture series, and it's called the Brent Eley Lecture Series. And it's named after a young boy, Brent Eley, who had a bone marrow transplant. And not here, he's from Colorado, but he was actually received his bone marrow transplant in Iowa. And his family realized, like, let's say 25 years ago, about how difficult it was to travel from out of state to another medical center to receive care. And so, unfortunately, he did not survive. But what his family did was create what's called Brent's Place, which is on the corner of the campus. And really what it is it's, I think, 68 rooms of different sizes, and it's a housing for kids and adults who need bone marrow transplant and solid organ transplant. And so they've sponsored this lecture for many, many years at this point. And in our lecture today is part of a growing list of real leaders in our field. And so what we do is we perform a special type of torture for this speaker, where they have to not only give cancer center lecture, which is the scientific lecture, but then a general pediatrics grand round lecture as well. And giving two different talks to two different audiences is really hard. And so thank you. And so our lecturer, our Brent Eley lecturer this year is Steve Dow. And Steve and I got to know each other probably about 5 years ago when my dog was diagnosed with osteosarcoma, and participated in some of the research that Steve will talk about. And that led to a friendship and a collaboration that's still ongoing, where we're trying to bring CAR T cells to dogs with cancer. Steve did his vet school training at the University of Georgia. He did his clinical work and postdoc training at CSU and at National Jewish, and he has been a leader in the vet space since then. So we're really looking forward to your talk, Steve. Thanks so much for coming and putting up with all our torture. My pleasure. Thank you, Mike, and thanks for the great opportunity to come down, reconnect with old friends. And really, I think one of the cement the bond in the vet school been really important. Many areas of endeavor for the past 20 years. Great opportunity to come and talk a little bit about what we're doing. On the drive down, I was thinking about the program and how long we've been working in this field. We've been basically working in this purpose drug platform for Myeloid for 5 or 6 years now. I think in the last years it's really begun. Lives begin to really out now. This is why we've been very fortunate to have a colleague of mine in leading. We've got part of this program and several of these collaborations with them. I don't think your microphone is working, so people are texting me. Maybe you could talk into this microphone. I think this is the only one that's connected to Zoom. Okay. All right. Is that better? Yeah. Okay. I think they're saying better. Good. Okay. No moving around. So Dan's a veterinarian, a good colleague of mine, a colleague of your team here. So Dan's bringing a new dimension, but I think the other reasons why we're starting to see, I guess, breakthroughs, but certainly new understandings, is we've actually had time now to enroll a lot more dogs in these trials. So the studies are maturing. We have new cohorts. I think the next thing we're beginning to realize is that this is a platform, and we're thinking about how better to apply it in combination therapy. So part of what I'll talk about this afternoon will be combining this with radiation therapy, with CAR T cell therapy, with Project with Mike,
combining it with other modalities, other drugs that are being repurposed, and more now targeted drugs. And then I think finally the big thing for us, and I guess is for me as an investigator, been the most exciting is that we've really begun to utilize next gen sequencing tools. So, we were somewhat stymied up until the last couple of years in terms of reagents and molecular tools, but really now sequencing, particularly focusing on the transcriptome, immune landscape, has, I think, closed a big gap that existed. So half of what I'm going to talk about this afternoon will be results from that sequencing work. And we'll be mainly talking about unpublished data, some data that's recently published, but we'll kind of walk you through the platform and then how we're working on making it better. These are disclosures. None of the disclosed efforts really impact on the work I'll talk about today. So to pose the question, I know a lot of the audience already understands why we do work in dogs and the value, but just for the rest of the group, reasons that we think the dog studies can be hugely impactful are, first of all, it's kind of validation, proof of concept of what we've learned in rodent models. And realistically, we won't be able to do the mechanistic studies in dogs to the same extent as we can in rodents. And we do a lot of work in rodent models as well, but it kind of takes it that next step and ultimately de-risk the human trials. And, Mike and I have had lots of discussions about why doing a CAR T cell study in dogs before the human trials would make sense. So there's that aspect. Dogs are similar in body size to us. Their metabolism is similar. GI tracts are much more like human than a mouse who's a hind gut fermenter. So that also impacts on toxicity. In the immune field, and I'm going to address this in the next slide, I think the dog's immune system offers a lot more insights than what we can always glean from mice. So we'll talk about why that is. And then for the dog lovers here, the story of kids and dogs and cancer, it resonates on many levels with many people. So it's always very rewarding for us to be able to think that the work is going to benefit both populations. So the dog immune system. So, I've had some time to think about this. So the dog immune system is much more educated than the mouse immune system. So as puppies, all of our dogs get vaccinated. They often get infections as young dogs, GI, respiratory. So by the time they've matured to get cancer, their immune system is nothing like the puppy immune system. And the mouse immune system really doesn't get that chance to become educated. We don't vaccinate mice. We have clean facilities, drinking water, food is all sterilized. So the different immune system, the dog also co-evolves with the tumor. We don't really know how long tumors exist in dogs before we detect them, but we guess it's months to even years. So the tumor and the dog immune system co-evolve. And then lastly, and I think this is a really important area that we're beginning to think about is, our dogs that get cancer are old. They may be 4 or 5 years old to us, but that's old in dog years. And they have an old immune system. And so that is also a variable that we often don't take into account in rodent studies. So the educated, vaccinated dog immune system that's aging can be, I think, a really good tool for really gaining some of these insights. So I started off by saying we're really excited about the new molecular tools, and for our lab, a lot of this is driven by a new grad student in the lab, Dylan Ammons, who's in our combined DV and PhD program. And Dylan has really done an amazing job to drive this technology for us. So one of the things that Dylan has done is he's created now an atlas, single cell atlas of dog circulating leukocytes. And he's carefully annotated the atlas. Spent a lot of time really building a good tool for us we can use this tool now to, for example, identify new markers for all the major dog leukocyte subsets. If you strip out granulocytes, which is part of this atlas, which is not typically present in most human PBMC atlases, the subsets are about equivalent, about roughly 30 to 32 primary leukocyte subsets, including T cells, monocytes, NK cells, B cells. So at that level, there's similarities, and we're exploring also at the functional level how similar or dissimilar the immune system is. But the point is, we have a tool now that we can use to address some really important questions. And one of the questions will be, how does the dog immune system respond to the presence of cancer? You know, we've really never had the tools before now to address that question. The things I'm going to cover, I'm going to talk about the platform, but what we've done to build on it. So looking at combinations, how they're working clinically, mechanistic studies on how they're impacting the tumor microenvironment. We'll look at taking the platform and applying it into two different tumors we haven't really talked about here. So in addition to osteosarcoma, we're doing studies in brain cancer in dogs. So, you know, I think you could argue the two best dog models for pediatric and human cancer are going to be bone and brain. So the brain tumor models, I think, instructive. And then a new model for us that we're really beginning to build on is cytonasal cancer. So dogs have long noses, so they actually unfortunately get a lot of nasal adenocarcinoma. It's not virally induced. We don't really know what triggers it, but it's a good model for a fairly rare human cancer, which is analogous, same location, same basic histiotype. So we're using the dogs to model the myeloid targeted platform with radiation therapy. So those will be the three themes for the afternoon. Just get everybody on the same page. These are the myeloid cells that we're targeting. They're all highly immune suppressive in cancer. So we're going to be looking at targeting macrophages, myeloid-derived suppressor cells, sort of this odd, ill-defined population of immature monocytes and neutrophils, and then really begin to focus on the neutrophils as well, in large part because of the IL-8 story. And I know that's becoming an important story in cancer, and it's part of Mike's story with the CAR T-cell targeting. So IL-8 and neutrophils is really the new target for us. You know, it all started with repurposed drugs, and Dan Regan in my lab really moved this work forward. He's been instrumental in getting it through the dog trials and into pediatric trials. So we took two drugs that we repurposed. Losartan, which is an antihypertensive drug, happens to have this really interesting off-target property. So it actually, in addition to its angiotensin receptor, also blocks signaling by the CCR2 molecule on monocytes. So it's a way to deplete macrophages by attrition. If you can block the recruitment over several weeks, the tumor will become depleted of macrophages. And it's a drug that's safe, it's easy to dose, and it's cheap. So we can do dog trials with it. The second drug is a homologue of sunitinib. It's an early generation, very non-specific tyrosine kinase inhibitor. Tosiranib, it's for osteosarcoma, it has no activity. It's ineffective, but for the immune system, it is actually highly effective. And it, for example, is an important drug for blocking STAT3 signaling. So Tosiranib and Losartan started it off. That was the platform. The new work now is adding a targeted drug called latorixin, which blocks signaling by the CXCR1 and 2 receptors, which is the IL-8 signaling pathway for neutrophils. So our basic approach is to screen the drugs in dogs with metastatic cancer. And I've just illustrated here, these are people's pets. So people that own large breed dogs kind of dread this tumor. It's unfortunately very common in large breed dogs. You know, Mike can speak from experience. We certainly see lots of these patients at the vet school. So these are dogs that have failed conventional cytotoxic chemotherapy. Most of them have been amputated because we don't really have a good way of controlling the primary tumor. So amputation, cytotoxic chemo, and then they enter our trials. And this is where we're trialing our immunotherapy combinations. So this was published last year, but this is kind of a summary of the work that built this platform. So it's losartan and tisiranib. Just showing you, first of all, that tisiranib has essentially zero activity as a sole agent. Again, dogs with metastatic chemotherapy-resistant cancer. The combination of losartan at the conventional dose, the standard dose, with tisiranib really begins to show a little activity, but essentially, really nothing to get that excited about. It was really only when Dan did the PK work to see what dose of losartan we actually needed to reach the effective therapeutic concentrations that we really stepped the dose up. So this is 10 times the conventional dose that we would use in internal medicine, for example, for hypertension in dogs. So big dose, fortunately, it's very well tolerated, really no AEs with respect to blood pressure or other sequelae from high dose losartan. So well tolerated drug combined with with tisiranib. So that combination now starts to show activity, right? So we see partial regression in 25% of treated dogs. And I should point out that metastatic osteo in dogs is pretty much the end. Conventionally, there's nothing we really have shown as active. So showing even some activity in this setting for us was a win. It's biological activity, so stopping the tumor from growing plus getting regression was 50%, which is, again, a big improvement over what we've had in the past. So this work has led to a pediatric trial here with Kelly Falk and Meg. Sorry, I'm blanking on Meg's last name, and Leah Gore kind of set it all up. But I will point out that in the pediatric trial, we're still at that standard dose losartan threshold. So it's an escalation trial. We're in the second cohort, and Dan is really instrumental on the back end of that for PK monitoring. So still not at the dose we'd like to be. And all that being said, that's probably why we're not really seeing much in terms of responses yet. But it's a platform. I think what we're beginning to rethink is maybe how to deploy that platform in a better way. Like, it may not be realistic to think that the platform is going to be that effective by itself, but so where is it that it can work? And that's, I think, where we're going to see better outcomes. One of the things we did was add in a third targeted drug. So Linda, my lab, has done a lot of the analysis for biomarkers in these studies. So we compared metastatic tumors in dogs to the primary tumors in the bone, looking at amputated samples, and sequence them. And what we found is that really this IL-8, CXCL8, CR1-2 pathway really pops up is one of the most upregulated pathways in the metastatic tumors compared to the primary. So it makes it an attractive target. And fortunately, we have a drug to target it with our colleagues at Tufts University working with Dompe Pharmaceuticals. They have a targeted drug called latorixin. The PK had already been worked out in dogs, good safety profile. So this was the drug we added in then to the tisiranib losartan backbone. So now when you add this third drug in, we start to see continued improvement. The thing that is really exciting is we now see complete regression. So 2 out of 20 dogs, so 10%, have had the tumors go away. One dog is out 2.5 years, so probably a full CR. And then we still see the same partial response rates, but the biologic response now jumps up to 70%. So it's incremental improvement, right? You're stacking complementary pathways, but every increment is a win. So we think this is an interesting platform, and this is only immunotherapy, right? So they're not getting any other treatment. They're done with chemo, no radiation, just oral medications. And so we're super excited about this particular pathway. The, if you look at survival, or the outcomes, we gain roughly 10 days. That's not a huge win. But we think what this teaches is where this would go in an adjuvant setting. So we've taken this data, proof of concept, to now move it in really where we want to be, which is to give the drugs up front at the time of amputation to block the metastatic pathway altogether. And this just is showing, here's one of our long-term survivors. This dog is out 2.5 years now. So multiple nodules regressed with only the myeloid targeted immunotherapy. So the trial right now is underway for this adjuvant protocol. So it's the same 3 drugs, same dosing. The drugs start 2 weeks before amputation. So the dogs come in to get staged. They start on the drugs, and then 2 weeks later they get amputated. And this is an immuno-only study, right? So there's no cytotoxic chemo in this particular trial. So for us, being as good as cytotoxic chemo would essentially be a win because it's oral only. We can give this long term. It's well tolerated. So the comparison here is with a big dog trial run 2 years ago, giving carboplatin, which is our standard therapy, with rapamycin. That trial showed no activity from the rapa, but it's a point of reference for survival and progression data. So on the right is the historical control, recent cohort. On the left are our dogs. We've enrolled 27 now. So the median progression-free interval is looking much better than carbo alone, and then the median survival, we've not reached the midpoint yet. So, it looks very promising. Again, probably isn't going to be the final answer, but it's certainly a step forward if this 3-drug oral combo can be combined with other conventional therapies. And then Linda looked at what correlated with survival in these dogs and improved survival. It's really interferon-related genes. It's an interesting set of genes that come out of this. It's not really what we would have predicted, but it certainly would speak to possibly T cell or NK cell mediated activity with interferon gamma or type 1 interferons. And then the top upregulated gene is a chemokine. It's not in the IL-8 pathway. It's a monocyte chemoattractant protein, and it may be a compensatory response to blocking the monocyte recruitment with the drug losartan. So back then to Dylan's work. So the study that we're about to submit, hopefully tomorrow, has taken the atlas from 7 healthy dogs. So these were adult dogs, same age roughly as dogs that develop osteosarcoma. No overlap of breeds, tried to space out the genders. And that atlas then is compared to 10 dogs with primary osteosarcoma who were, the blood was drawn before they got any treatment. So osteosarcoma naive, primary tumors, and then really just comparing the transcriptome response in circulating leukocytes. So so what comes out of this is some unexpected findings for us. We had really expected to see a lot of changes in the T cells, perhaps the NK cells. Weren't sure about the B cells. Jill might be able to weigh in there, but the things that really stood out was that the changes that occur, again, in dogs with primary osteosarcoma, the changes that are occurring in the myeloid cell population. So in the monocytes, in the neutrophils, and to some degree in the circulating dendritic cells. It's not to say there are not any T cell responses or changes. There are some. There's a dropout of double negative T cells. There's an upregulation of CCL5 expression on CD8 effector T cells. So there are changes, and we actually lose a lot of circulating B cells in the dogs with cancer. So the changes occur, but they really seem to be focused on the myeloid cells, which, I think kind of helps build a rationale for why targeting these cells makes sense. And then the latest work, and it's still being analyzed, is to take now tumor biopsy samples. So these are again from primary tumors in dogs in the adjuvant trial. So they get a tumor biopsy before any treatment, and then they get a biopsy 14 days later on the triple drug cocktail. And we take 5 samples from each tumor biopsy to make sure we're sampling accurately or covering the tumor, pool the samples, single cell sequencing. Dylan has analyzed 75,000 cells for this analysis. And then really asking, what are the changes in the leukocyte infiltrating cells? And then what are the drugs doing to these cell populations? So the analysis is not complete, but there is a story around myeloid cells, which I'll show you in a second. But it is interesting to look at the tumor microenvironment in dogs with osteo. It is heavily infiltrated primarily with myeloid cells. There are some T cells, and then there's a lot of fibroblastic component, which speaks to why, for example, a FAP-targeted CAR T cell makes sense. There's a nice tight tumor cell population. There's an osteoclast population because it's coming from bone. So it's a good representation of a fairly complex environment. But what comes out of the early analysis and the question here is just what are the triple drug cocktail doing? It's really primarily depleting myeloid cells. So that's the cells that are being lost in the dogs on 14 days of therapy. There are some other cell changes that occur as well, including in T cells. And this is 14 days, and we don't really know what would happen after 30 or 60 or 90 days of treatment, but it is a look at the early responses to this cocktail. So I guess before we transition to the next part of this, kind of take-home messages, it kind of makes sense, right, that targeting multiple pathways would give you a better outcome as long as they're complementary and not antagonistic. Myeloid cells seem to be the main target in animals with cancer in terms of the circulating immune system being aware of the tumor. And then the triple drug cocktail really seems to target the myeloid cells in that tumor microenvironment. It actually is remarkably homologous. So there are plasmacytoid DCs or myeloid DCs. So it's a very good question. We really just haven't drilled down yet on the tumor DC population, but from leukocyte analysis, we do have a tool now to see where the normal cells what their function would be in their relative abundance. So I'll repeat Mike's question for the home audience, but Mike is asking about a CDC1 positive dendritic cell population having prognostic significance in osteosarcoma. And we just haven't looked. So we're sitting on a bunch of data. And we're just trying to get through it right now. But the second paper would be on that sort of response, right? In osteosarcoma, just in comparison to the circulating cells. And we do have outcome data on the dogs, so we could begin to make those correlations. Yeah, so Mike asked, do the dogs stay on drug? And the answer is yes. And it's essentially going to be lifelong. Like we don't really have any plans to stop. We can certainly get losartan and tocitinib forever. Latorixin's a little trickier because it's coming from a biotech, but for the trial, 6 months is our cutoff for latorixin. but we would likely continue for any surviving dogs with at least losartan and tacrolimus. Yeah, so it's continuous, and I think that's how we would look at it, right? This is a long-term strategy. It's not just not one and done. Yeah, so Lanarixin is provided free by Dompe. You know, their target is not cancer; it's really type 1 diabetes and pancreatitis because of the inflammatory component. We've talked to them about an interest in this, and they seem interested, but right now, but Reparixin is on the market. So there is a drug very similar to this that could be repurposed. Yes. The question would be the side effect. Is there anything that becomes more toxic with adding Lanarixin? And I guess the only thing we have seen is wound healing. So we're actually in not surgical site infection. So we have had 2 dogs that have had surgical site infections where we've had to stop the drug to try and resolve it, probably because of inhibiting neutrophil migration to the infection site. But otherwise, the drug combination is essentially the same as the double drug in terms of side effect profile.
As the dog owner who had this, diarrhea is a major issue. It's not a huge issue, but it's just
No, no, it's a good point. I mean, the GI tract is the dog major toxicity organ, so I think it does predict GI toxicity really well, and that's mainly a tacrolimus side effect. It's from the targeted the TKI drug. So there are 3 studies I'm going to talk about now. So again, taking the platform, myeloid targeted therapy, off-the-shelf drugs, and then now looking at them in combination with other modalities. So the first trial, and this was published last month, is in a dog glioma brain cancer model. Glioma is very analogous to pediatric brain cancer. It's not quite as genetically aligned with adult brain cancer, but very good model for studying pediatric brain cancer. And I'm going to come to propranolol now because I know it just kind of parachuted in, but this 2-drug combination, losartan and propranolol, we chose for brain because of CNS penetration. So both drugs are highly penetrant into the CNS and CSF. So that's why propranolol over tacrolimus. It's also a very interesting immune modulatory drug, what I'll get to in a second. Canine glioma, but compared it with a cancer stem cell targeted vaccine. So you can take tumor cell lines, not allow them to attach, take away their serum, put in a replacement cocktail of cytokines and growth factors, and they will dramatically change their stem cell marker profile, and they'll markedly upregulate stem cell antigens. So we've taken dog tumor cells grown under these spheroid conditions, made a vaccine with a very effective adjuvant for cross-priming, and given that now with the losartan propranolol combo. Second trial is a radiation trial in dogs with sinonasal cancer. It's a randomized trial. They get conventional radiation, or they get radiation plus losartan propranolol. Really interesting results coming out of that trial, which I'll get to. And then the last study with Mike and Jessica is going to be the B7H3 targeted CAR T cells in dog osteosarcoma with a slightly different platform. It's losartan and propranolol rather than losartan and tacrolimus. So, where did propranolol come from? So some really interesting studies done years ago found that mice housed at what is comfortable to us room temperature is highly stressful. They're very cold at 72 degrees. They'd rather be at 80. And so investigators at Roswell Park and other institutions made this really interesting observation. If you raise the body temperature, the the housing temperature for mice, their tumors are much better controlled. So working through that observation, they've made the connection with catecholamine release from thermal stress, and then realized that you could block that catecholamine response with a beta blocker like propranolol. And it's very interesting. It's really only propranolol that works. The better beta blockers and more selective blockers, you lose that effect from propranolol. So propranolol is a dirty blocker. It blocks beta 1 and beta 2, and that may be part of why it's more efficacious. So that's the drug we've opted to use, and we have some preliminary data from rodent models, make sure it made sense, the combo. You get an additive effect. It doesn't cure the tumors in mice, but it does knock their growth down. But what's really interesting is when you look at the TIL population in these combo treated mice, you get this infiltrate of CD8 T cells and to some degree CD4. And it's only seen with the propranolol combination, not with carvedilol, which is a more selective beta blocker. So, but that combination seems to be uniquely able to recruit T cells into the tumor, which you don't see with either drug alone. And we've modeled this in human leukocyte cultures and in dog in vitro cultures as well. It's interesting, these drugs at least in vitro, don't really seem to affect that many immune pathways. It's really these metabolic pathways that are affected, particularly by propranolol. And then when you put the two together, you get this whole new set of genes that are altered. So it's not entirely clear in this context how that combination works. And you could argue, well, this is an imperfect way to model it, but at least it's a start. So point being is that combination has some unique properties. So the first trials was just published last month. Dogs with gliomas, a small trial, 10 dogs. In our patient population, you have basically two options. If your dog gets a brain tumor, you can have surgery or you can have radiation therapy. There really isn't, we don't have targeted drugs. Temozolomide doesn't work very well in dogs, so it's really radiation or surgery. So the study we did was with Rebecca Packer. She's a neurologist at the vet school. Dylan was involved in the back-end analysis of the data. It's a stem cell targeted vaccine. The vaccine is given at twice weekly intervals for 3 months, and then it's spaced out to monthly for a year. And they're on continuous oral losartan and propranolol dosing. So those two drugs never go away. So what we saw, so again, small study, but again, these dogs get nothing, they're getting no other treatment. So no debulking, no RT, it's only oral immuno with the vaccine. 2 of the dogs experienced significant partial regression, illustrated on the right. With top is the pre-treatment, and the bottom is MR after treatment. So significant and durable tumor regression, so that these responses last. Other dogs have had tumors essentially stop growing. So if you look at our biological response rate of PRs, partial response, and stable disease, it's almost 80% of these dogs. So it's a big biological readout. The responses almost entirely correlate with responses to the vaccine. So I don't think so the 2 drugs are really setting the stage for the vaccine to work. In rodent modeling, we've been doing the 2 drugs are active, but not nearly as active as they are with the vaccine. So dogs that responded to the vaccine had tumor control. Dogs that didn't control the tumor. So where these studies are going, they're really sort of foundational work to apply this in a setting where it would make sense in an adult or pediatric population. So the next study we're starting now is going to be combining this with radiation therapy. So SBRT, targeted therapy to the brain. And this intersects with discussions Mike and I have had, where do you give the radiation? And that's what we're trying to figure out in rodent modeling. Is it better to give radiation up front, give radiation at the time you start immuno, or actually delay radiation until after? And the early data is quite intriguing. It looks like delaying may actually be the better option, which doesn't make a lot of sense. And you can come up with an explanation, but it wasn't what we were expecting. So that's where the trials now in glioma are going, again, building off the platform. Any questions on brain? Yeah, so Mike asked the same dose of losartan, and yes, it is the high dose losartan. We are more careful with propranolol, so it is ramped up gradually. And that there are, we've had a couple of instances where we've had to back off the propranolol dose. Just it is a pretty vasoactive drug. We've kept the losartan constant though, and that generally solves the. It's been weakness, lethargy, anorexia, kind of vague signs, but no overt hypotension. So the next study is this RT study. So nasal cancer in dogs, very common, almost like bone cancer. It's a very common tumor. Interestingly, it's only in dogs with long noses. We don't see it in boxers and pugs and Bostons, but it's common, very difficult to treat. So Kara's boss has been really heading up these studies. She's a rad onc doc at that school. Thomas Lee is her post-DVM fellow. And then we've been working with Sana Karam here at the medical school. And this is Kara's story, so I'm not going to say much about it except to look at the immune response. So we've come up with, I think, was a fairly clever solution to try and get biopsies from these samples to figure out how to monitor, to do immune monitoring. It's very difficult to biopsy these tumors, even rhinoscopically. You just can't get good tissues. It's invasive. It's an anesthesia. So what we worked out was to pass a soft rubber red catheter into the nasal cavity and just flush in warm saline. And if you do that 2 or 3 times, you actually get a nice sampling of the immune cells in the nasal cavity and around the tumor. So we've been using those samples then to monitor treatment responses or the immune response. So what we found is this really interesting phenomenon. So the first couple of months after radiation, it's not much is going on. There's a spike at 24 hours of inflammatory cells, and it kind of quiets back down. But then, really, at 3 months is where it gets interesting, and we see this phenomenon where you get this upregulation of chemokine production, particularly IL-8 and MCP. Those are the chemokines that are going to recruit the neutrophils and the macrophages into the tumor, and also IL-6. And interestingly, so the dogs in blue are the radiation only, dogs in red are radiation plus propranolol losartan. When you add in the 2 drugs, and this is a randomized trial, we really knock down that chemokine production, which is not exactly what we'd expected, but we'll take it because it kind of makes sense for what we're trying to do to modulate that myeloid response. These tumors are heavily infiltrated with macrophages. Radiation makes that worse. Kara published this work on a different tumor last year. So radiation definitely triggers an inflammatory response. The next data for the T cell people is actually, I think, even more interesting. So again, using lavage samples, quantitating T cells, and we're doing single cell on these samples as well as bulk sequencing. But by flow, again, at 3 months, you get this phenomenon where the tumors begin to become infiltrated or the nasal cavity with particularly CD8 T cells, but it's only in the combination treated group, not in the SBRT alone group. It seemed the same thing with CD4 T cells. And what we did not see was it didn't really block the monocyte recruitment. It kind of stayed the same. But, again, getting recruitment of T cells kind of harkens back to that mouse study I showed you where we had the TIL infiltrates in the tumors of the mice getting the combo, again, the losartan propranolol combo. I don't really understand this phenomenon well at all, but it is an interesting phenomenon. And Kara's got clinical data, which she'll present actually at Tumor Journal Club soon, but it is correlating with clinical responses as well. So it's nice to see the immune correlates with clinical outcomes. And then finally, kind of where I started with Mike, I don't remember exactly where this even started. Well, it was getting your dog Sadie enrolled in the trial. So fast forward to today, Mike's obviously got a long-standing interest in CAR T cells. He's really done a lot of nice work with B7 targeted CAR T cells. So B7H3 is a checkpoint molecule expressed primarily on myeloid cells, but highly overexpressed on certain tumors in dogs and humans, including osteosarcoma and glioma. So this is a trial with Jenna Cow in my lab. She's another DVM PhD student. Mike and Jessica are running things from here at the medical school. So it's a V Foundation funded grant to look at B7H3 CAR T cells in metastatic osteo. and where, again, the myeloid part of this is we're going to use the losartan propranolol combo. We opted not to use tesserative, partly for tolerability. It's just a lot easier to dose losartan and propranolol. And then also because of this CD8 T cell effect that we had seen. So we're building dog B7 CAR T cells. Jenna has got the magic touch. She trained in Phil Greenberg's lab, so she knows her way around culturing T cells. So Jenna's done a really nice body of work to look at the expression of B7H3. It's highly expressed in osteosarcoma in dogs. We don't really find expression anywhere in normal tissues. And so it's a good CAR target. And then she's shown that the dog CAR T cells will kill dog osteosarcoma cell lines. And it's the killing and the cytokine response is actually proportional to the amount of B7 expressed. So, it's a nice biological readout of the T cell in interaction with its cognate ligand on the tumor cell. And Jenna's done a lot of work, which won't really show up in her publication, but to optimize CAR T generation and then to show the activity in killing assays. So this kind of built the foundation then for the work we're doing with Mike. We all know that the field right now struggles in solid cancers to get CAR Ts to work. A lot of people in this audience working on it. Multiple strategies. The two that we're using concurrently is to get the T cells into the tumor better, so trying to solve the trafficking issue, and then looking at modeling, remodeling that tumor immune suppressive microenvironment. And so our approach has been to use the CAR that Mike developed. So it's a dual CAR. It's a B7 targeted CAR that also expresses the ligand CXCR2 for IL-8. It's not attached to the CAR. It's expressed separately in the same cell. And the idea, which is very clever, we'll give Mike credit for that, is that many tumors overexpress IL-8, and particularly after you irradiate the tumors, that expression goes up even higher. So it's a good strategy to get the CARs into the tumor, relying on that chemokine gradient. And Jessica has shown a really nice work that the dual CAR compared to the single CAR, compared to just B7 CAR, really looks much more promising in the rodent models. The single CAR overlaps with the control mice. The dual CAR essentially clears the tumor. So we've taken the same CAR, put it into dog T cells, and then run it through our dog xenograft model. So it's skid beige mice with the dog osteosarcoma tumor, where we infuse the dog CAR T cells. So the early generation CAR, single CAR, didn't do a whole lot, but it's kind of what we'd predicted from these solid tumor models in mice. The dual CAR is better. It's not as good as what Mike and Jessica see, but it's a different mouse model. And it's also a canine tumor, not a human tumor. And I think the human tumor in those models is engineered to overexpress IL-8. So there's some differences in the model, but it is still better than the single. And so the plan at this point is that this would go into trials in dogs with metastatic osteosarcoma. So we've gotten the approvals. We know how to generate the CARs. They would get treated with the oral losartan propranolol cocktail. And our readout is going to primarily be tumor responses. We'll also try and track the CARs in the blood as well. So that's the story. We have many projects going on. It's been great to collaborate with folks here at the medical school, with Mike and others, and Jill. But I guess our story right now is that you can actually control, we're not curing, but controlling metastatic disease with just oral myeloid targeted therapy, which I think is a significant advance. Stacking the drugs works better. It's incremental. I think the challenge is to come up with the best stack. Pretty much our work is going to build off of that platform of losartan and something. It's either going to be propranolol or docernib. It's the myeloid cells that sense the tumor, at least in blood. And the myeloid cells are the target of the triple immunotherapy. And again, kind of back to where we started, it's basically a versatile platform to build other approaches on top of. And that's, where we'll be putting our efforts for the next foreseeable future. So again, pleased to be invited here. It's great to catch up with folks. Happy to entertain any questions you might have. I think that CARs just kind of stop working. I mean, they're very hard to find in the tumors. So I don't think that there are many actually getting there. So I don't really know fully how to account for that. Mike, you want to speculate, or others?
I'm not sure our model is very binary. You're totally clear as a tumor, large expansion or I'm not sure. Is that single? Okay, and are those immune competent models?
These are immune deficient. The difference in our models and Mike's models, our mice have myeloid cells, which we need for the myeloid targeted component of the study. I think your mice, I thought they're NSG mice, they have some, yeah. And these are not with the 2-drug combo. These are mice with just CARs alone. There's some issues with the 2-drug combo that we're trying to work through. It's a really good question, good point, actually. We have the tumors, we just haven't gotten to processing them through, but it's something we probably should go back and look at. We've kind of been focused right now on getting the 2 drugs to work best with the CARs, and then kind of figure it out after that.
Like I said, we've looked and we haven't seen it. I think Ross might have a question.
Did someone just say, did Ross have a question? Excellent.
Hey, Ross.
Good to see you, man. Sorry I'm not there.
Yeah.
I'm to miss you.
No worries.
So Kim Rathmel had some really interesting data, maybe a couple years ago now, on the fact that tumor cells actually aren't competing much for glucose. They actually really like glutamine. And it's almost exclusively the myeloid cells that are really addicted to glucose. Actually, T cells, I think lymphocytes were much more glutaminolysis related as well. So you had the first study, you had rapamycin, and you were kind of looking at myeloid populations, and it would make some sense that you might negatively affect those. Is there, in terms of tumor regression, have you ever thought of like trying to inhibit glutaminolysis or glutamine uptake inhibitors or anything along those lines, and couple that with either rapamycin or other of your combinations?
Yeah, so Ross's question is about targeting glucose uptake. And so our lab hasn't looked at this, but Doug Tham's lab at the Animal Cancer Center has done some rodent work with it. We haven't done anything in dogs. It takes a lot of the inhibitor. I guess that's part of it. We don't really understand how to dose it and off-target effects, but it's a strategy. We haven't really swung back to it, but it might be something to consider, particularly in combination. I forgot acknowledgement, sorry. So, folks in my lab that have worked on the project, Dan Regan and his group, obviously Mike here, Children's and Anschutz, and then our colleagues at Tufts as well, and then our support. Yeah.
Models where you've inhibited the myeloid tumor microenvironment, are you seeing more infiltrating lymphocytes? Could those either be giving you or a path where you could start to augment the T cell part of the immune system, get stimulated, and then see if you can augment them, preferably in vivo, but even taking them out and going back to the old still experiments where you augment them ex vivo. They were instructive experiments, obviously.
Yeah. So, yes, thank you for asking that. So it's interesting. We have not really appreciated a big T cell infiltrate in rodent studies or in dog studies in the dogs getting losartan treatment. And in fact, in that triple drug study, the early single cell data looks like there's actually a depletion of T cells. So so the mechanism, it still may involve T cells, but it certainly does involve a lot of them moving into the tumor. So it's a bit of a disconnect. We still think it's a T cell mediated response, but we don't know exactly how to explain that the T cells are not there where they should be. They're there with propranolol, but not with. Yes, it could, right? We would expect to see that in the mouse model.
Although I'll say Rob's question and Jill's question kind of makes me wonder a little bit, like, are these antigen-specific cells, and these cells, and then see whether they're really tumor reactive, and using other methodology to really know whether they're just kind of occupying space that they could not occupy because the myeloid cells were there, or are they really like antigen specific and they're expanding within the tumor at having a response or potential response?
It's something that it bears looking into for sure. We've not gone there yet, but I think we're getting the tools now to start to do some of that bioinformatically. Yeah.
In fact, some of those RNA-seq methods, you could also
Right.
add TCR to it and
Yeah.
understand formality and things like that. So
And we are starting to look at that actually. Yeah. Yeah, so we're looking at the tumor separately. It's just not in the analysis today. In the tumor, I will let you know. Yeah. Historically, that tumor doesn't express the MHC class 2. You know, it's a really cold, dreary tumor, you know, just not much going on. Not like carcinomas, yeah.
Positive therapeutic benefit. A lot of evidence that the myeloid cells that you're suppressing are actually producing is augmenting the tumor's growth, and you're removing the growth factor. It's not the way we think, but we think that we're removing something that suppresses the immune response.
Yeah, I mean, there's pretty good evidence that the myeloid cells will release trophic factors for the tumor as well. Particularly angiogenic factors, matrix metalloproteinases promote extravasation and metastasis. So yeah, the tumor benefits from those cells being there, even without T cells. Okay.
Thank you.
Thank you.